Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. In this lecture, we will be seeing development in food microbiology and current status. In this lecture, we will be covering the development in food microbiology, and in this basically we will be seeing the history and development of the food microbiology. And in this, let us start with this milestones in food preservation, then milestones in food fermentation, then milestones in foodborne poisoning, milestones in microbiology techniques, milestones in food legislation, and then food microbiology we will be seeing the current status, and among the current status the food fermentation or the probiotics, food spoilage, foodborne diseases, and some miscellaneous things we will be discussing. So, let us start with the development in food microbiology. The major developments of ideas on the possible roles of microorganisms in foods and their scientific proof were initiated by Pasteur in the 1870s, followed by many other scientists before the end of the 19th century. This paved the way for the establishment of early food microbiology in the 20th century. Milestones in food spoilage. In 1659, Kircher demonstrated the occurrence of bacteria in milk. Bondieu did the same in 1847. In 1780, Scheele identified lactic acid as the principal acid in sour milk. In 1804, François Nicolas Appert developed methods to preserve foods in sealed glass bottles by heat in boiling water. In 1819, Peter Durand developed canning preservation of foods in steel cans. Charles Mitchell introduced tin lining of metal cans in 1839. In 1857, Pasteur showed that the souring of milk was caused by the growth of organisms in it. In 1867, Martin advanced the theory that cheese ripening was similar to alcoholic, lactic, and butyric fermentations. In 1870, Louis Pasteur recommended heating of wine at 62.7 degrees Celsius for 30 minutes to destroy souring bacteria. Later, this method was modified and named pasteurization and used to kill mainly vegetative pathogens and many spoilage bacteria. In 1873, the first reported study on the microbial deterioration of eggs was carried out by Guénon. Lister was first to isolate Lactococcus lactis in pure culture. In 1876, Tyndall observed that bacteria in decomposing substances were always traceable to air, substances, or containers. In 1887, Foster was the first to demonstrate the ability of pure cultures of bacteria to grow at 0 degrees Celsius. In 1895, Harry Russell showed the gaseous swelling with bad odors in canned peas was due to the growth of heat resistant bacteria, which was termed as spores later on. In 1915, Bacillus coagulans was first isolated from coagulated milk by B.W. Hammer. Now, the milestones in food preservation. In 1782, canning of vinegar was introduced by a Swedish chemist. In 1843, sterilization by steam was first attempted by I. Windsor in Maine. In 1845, S. Eliot introduced canning in Australia. In 1853, R. Chevalier obtained a patent for sterilization of food by autoclaving. In 1854, Pasteur showed heating to remove undesirable organisms was introduced commercially. In 1855, Greenwade in England was the first to produce powdered milk. In 1874, the first extensive use of ice in transporting meat at sea was begun. In 1878, the first successful cargo of frozen meat went from Australia to England. In 1880, the pasteurization of milk was begun in Germany. In 1890, commercial pasteurization of milk was begun in the United States. In 1908, sodium benzoate was given official sanction by the US as a preservative in certain foods. In 1928, the first commercial use of controlled atmosphere storage of apples was made in Europe. In 1929, a patent issued in France for the use of high energy radiation for the processing of foods. In 1954, the antibiotic Nissin was patented in England for use in certain processed cheeses. In 1988, Nissin was accorded GRAS, that is the generally recognized as safe status in United States. In 1990, irradiation of poultry was approved in the United States. In 1997, the irradiation of flesh beef up to a maximum level of 4.5 kg Gray and frozen beef up to 7 kg Gray was approved in the United States. Now the milestones in food fermentation. In 1822, C.J. Persson named the microscopic organism found on the surface of wine during vinegar production as Mycoderma mesentericum. Pasteur in 1868 proved that this organism was associated with the conversion of alcohol to acetic acid and named it Mycoderma aceti. In 1898, Martinus Beijerinck renamed it Acetobacter aceti. In 1837, Theodor Schwann named this organism involved in sugar fermentation as Saccharomyces, which is a sugar fungus. In 1838, Charles Cognard Latour suggested that growth of yeasts was associated with alcohol fermentation. In 1860, Louis Pasteur showed that fermentation of lactic acid and alcohol from sugar was the result of growth of specific bacteria and yeast respectively. In 1883, Emil Christian Hansen used pure cultures of yeast to ferment beer. Now the milestones in foodborne diseases. In 1820, Justin Kerner described food poisoning from eating blood sauces due to botulinum. Fatal disease from eating blood sauces was recognized as early as 900 AD. In 1849, John Snow suggested the spread of cholera through drinking water contaminated with sewage. In 1854, Filippo Fassini named the cholera bacilli as Vibrio cholera, which was isolated in pure form by Robert Koch in 1884. 1856, William Budd suggested that water contamination with feces from infected person spread typhoid fever and advocated the use of chlorine in water supply to overcome the problem. In 1885, Theodore Escherichs isolated Bacterium coli, later named Escherichia coli, from the feces and suggested that some strains were associated with infant diarrhea. In 1888, A. Gertner isolated bacterium which was later renamed as Salmonella enteritidis from the organs of a diseased man as well as from the meat the man ate. 1894, J. Dennis associated pyogenic staphylococcus with death of a person who ate meat prepared from a diseased cow. In 1895, Mary von Ermengem isolated Bacillus botulinum, which was Clostridium botulinum, from contaminated meat and proved that it caused botulinum. Now milestones in foodborne poisoning. In 1820, the German poet Justinus Kerner described sausage poisoning, which in all probability was botulinum, and its high fatality rate. In 1857, milk was incriminated as a transmitter of typhoid fever by W. Taylor of Penrith, England. In 1888, Gertner first isolated Salmonella enteritidis from meat that had caused 57 cases of food poisoning. In 1894, T. Dennis was the first to associate staphylococci with food poisoning. In 1896, von Ermengem first discovered Clostridium botulinum. In 1906, Bacillus cereus food poisoning was recognized. In 1926, the first report of food poisoning by streptococci was made by Linden, Turner and Thom. In 1938, outbreaks of Campylobacter enteritis were traced to milk in Illinois, USA. In 1939, gastroenteritis caused by Yersinia enterocolitica was first recognized. In 1951, Vibrio parahemolyticus was shown to be an agent of food poisoning by T. Fujino of Japan. In 1955, Scambroid which is a histamine associated poisoning, was recognized. In 1960, the production of aflatoxins by Aspergillus flavus was first reported. In 1965, food-borne giardiasis was recognized. In 1971, first US-born outbreak of Vibrio parahemolyticus gastroenteritis occurred in Maryland. In 1975, Salmonella enterotoxin was demonstrated by L.R. Koppel and R.H. Diebel. In 1976, first US foodborne outbreak of Yersinia enterocolitica gastroenteritis occurred in New York. In 1978, documented foodborne outbreak of gastroenteritis caused by the Norwalk virus occurred in Australia. In 1979, foodborne gastroenteritis caused by non-Vibrio cholera occurred in Florida. Earlier outbreaks occurred in Czechoslovakia in 1965 and Australia in 1973. In 1981, foodborne listeriosis outbreak was recognized in the United States. In 1982, the first outbreaks of foodborne hemorrhagic colitis occurred in the United States. In 1983, Campylobacter jejuni enterotoxin was described by Ruiz Palacios et al. In 1986, bovine spongiform encephalopathy was first diagnosed in cattle in the United Kingdom. Now some milestones in microbiology techniques. In 1854, Heinrich Schroeder and Theodore van Dusch used cotton to close tubes and flasks to prevent microbial contamination in heated culture broths. In 1876, Carl Wigert used methylene blue, which is a synthetic dye, to stain bacteria in aqueous suspensions. In 1877, Ferdinand Kohn showed heat resistance of Bacillus subtilis endospores. In 1878, Joseph Lister isolated Streptococcus, which is now known as Lactococcus lactis, in pure culture by serial dilution from sour milk. In 1880s, Robert Koch and his associates introduced many important methods that are used in all branches of microbiology to purify and enumerate bacteria. In 1884, Hans Christian Gram developed gram staining of bacterial cells. Now let us see some milestones in food legislations. In 1890, the first national meat inspection law was enacted. It required the inspection of meats for export only. In 1906, the US Federal Food and Drug Act was passed by Congress. In 1910, the New York City Board of Health issued an order requiring the pasteurization of milk. In 1939, the new Food, Drug and Cosmetic Act became law. In 1957, the US Compulsory Poultry and Poultry Products Law was enacted. In 1963, the US Food and Drug Administration approved the use of irradiation for the preservation of bacon. In 1967, the US Wholesome Meat Act was passed by Congress and enacted into law. In 1968, Food and Drug Administration withdrew its 1963 approval of irradiated bacon. The Poultry Inspection Bill was signed into law. In 1969, the US Food and Drug Administration established an allowable level of 20 ppb of aflatoxin for edible grains and nuts. In 1973, the state of Oregon adopted microbial standards for fresh and processed retail meat. They were repealed in 1977. Now, food microbiology current status. In the early 20th century, studies continued to understand the association and importance of microorganisms, especially pathogenic bacteria in food. Specific methods were developed for their isolation and identification. The importance of sanitation in the handling of food to reduce contamination by microorganisms was recognized. Specific methods were studied to prevent growth as well as to destroy the spoilage and pathogenic bacteria. There was also some interest to isolate beneficial bacteria associated with food fermentation, especially dairy fermentation, and study their characteristics. However, after the 1950s, food microbiology entered a new era. Availability of basic information on the physiological, biochemical, and biological characteristics of all diverse types of food, microbial interactions in food environments, and microbial physiology, biochemistry, genetics, and immunology has helped open new frontiers in food microbiology. The food fermentation or the production of probiotics. Development of strains with desirable metabolic activities by genetic transformation among strains. Development of bacteriophage resistant lactic acid bacteria. Metabolic engineering of strains for overproduction of desirable metabolites. Development of methods to use lactic acid bacteria to deliver immunity proteins. Sequencing genomes of important lactic acid bacteria and bacteriophages for better understanding of their characteristics. Food biopreservation with desirable bacteria and their antimicrobial metabolites. Understanding of important characteristics of probiotic bacteria and development of desirable strains. Effective methods to produce starter cultures for direct use in food processing. Now, the food spoilage. The identification and control of new spoilage bacteria associated with the current changes in the food processing and preservation methods. Spoilage due to bacterial enzymes of frozen and refrigerated foods. development of molecular methods, that is nanotechnology, to identify metabolites of spoilage bacteria and predict potential shelf life of foods. Importance of environmental stress on the resistance of spoilage bacteria to antimicrobial preservatives. Now let us discuss some foodborne diseases. The methods to detect emerging foodborne pathogenic bacteria from contaminated foods. Application of molecular biology techniques, that is nanotechnology, for rapid detection of pathogenic bacteria in food and environment. Effective detection and control methods of foodborne pathogenic viruses. Transmission potentials of prions diseases from food animals to humans. Importance of environmental stress on the detection and destruction of pathogens. Factors associated with the increase in antibiotic resistant pathogens in food. Adherence of foodborne pathogens on food and equipment surfaces. Mechanisms of pathogenicity of foodborne pathogens. Effective methods for epidemiology study of foodborne diseases. Control of pathogenic parasites in food. Now miscellaneous. Application of hazard analysis of critical control points, that is HACCP, in food production, processing and preservation. Novel food processing technologies. Microbiology of unprocessed and low heat processed ready to eat foods. Microbial control of foods from farm to table, that is total quality management. And lastly, the food safety legislations. Conclusions. The human civilization began when hunters and gatherers adopted not only production but also preservation of foods. Thus, long before the existence of microorganisms was discovered, their importance on food spoilage and health hazard was conceived by our early ancestors. Once their association and importance in food were proven, efforts were made to understand the basic principles associated with food and microbial interactions. This knowledge was used to control undesirable microbes and effectively use the desirable types. Current investigations are directed towards understanding microbes at the molecular level. A food microbiologist should have a good understanding of the historical as well as current developments in food microbiology, along with the characteristics of microorganisms important in food. That's all for today. Thank you very much.